In this last portion of today's lecture, we're going to take a look at the other ways that cells regulate the expression of their genes other than these global and fine control mechanisms over transcription. Remember, there's a long pathway between the information in DNA being used to produce an active protein. And that's what this slide is showing us. This is a representation of a eukaryotic cell. You can tell that because the artist has drawn in the nucleus over here, or at least what represents the inside of the nucleus, and the cytoplasm out here. Take a look at this pathway. Remember, DNA gets transcribed to produce RNA. RNA gets translated to produce protein. That's our central dogma. The third statement of the dogma tells us that protein creates traits. In reality, though, in cells that are as complex as eukaryotic cells are, those first two statements are not complete. In reality, the first thing that's produced through transcription is that immature RNA transcript. Remember, it has to be processed. The introns have to be spliced out so that the exons can join together. A cap has to be added, a tail has to be added so that you get a mature mRNA. That mRNA then has to move out of the nucleus through a pore and into the cytoplasm. These are times that a cell can intervene and stop the expression of a gene. In other words, even if transcription of a gene occurs and you get an immature transcript produced, that doesn't necessarily mean you're going to have an active protein produced. A cell can step in here and control whether or not splicing occurs, whether or not the RNA gets capped or gets a tail put on it, whether or not it gets transported out of the nucleus. The cell has control over all of these things. Even if the messenger RNA gets transported out into the cytoplasm, it doesn't necessarily mean that an active protein is going to be produced. Once it's in the cytoplasm, the cell has control over how long this messenger RNA is going to survive, how long before it's going to be degraded. Once the messenger RNA gets degraded, no protein can be made from it. Now, even if the messenger RNA were to get translated, the cell still has some control over whether or not an active protein is produced. Remember, translation produces a polypeptide. That polypeptide has to then be processed, folded, and altered before you get a protein. And just because a protein gets produced, it doesn't mean that protein is active. Cells have the ability to add small molecules onto proteins and inactivate them. So it's a long journey from the DNA that is a gene to the production of an active protein. And yes, a lot of the global and fine levels of control happen right here. But it's important to remember that cells have control at all of these places. And they can step in and prevent the production of active protein at any of those points. Now, interestingly enough, as if that isn't a complex regulatory control system, cells have additional levels of control over gene expression outside of the flow from DNA to active protein. Remember, we talked about these regulators and activators, and we said that those regulatory proteins can themselves be turned on and turned off. But there are additional molecules inside of living cells that can interfere and help regulate the expression of genes. We often refer to these additional molecules as signaling molecules. You'll hear that term a lot. What are these signaling molecules? Well, some good examples of these are hormones, different types of drugs or pharmaceutical products, lots and lots of toxic molecules like pesticides and herbicides and so on. In fact, most highly nonpolar molecules, highly lipid-soluble molecules, can act as signaling molecules. The reason that the lipid-soluble portion or part of this is important is because these are molecules, these signaling molecules, that are going to help control gene expression by coming into a cell. And as we now know, it's hard for molecules to come into a cell unless they meet certain chemical characteristics. Hormones and drugs and pesticides tend to be lipid soluble, and lipid soluble things get into cells more readily and therefore are better choices to use as regulators of gene expression. So yes, there are lots of mechanisms outside of transcription and translation that can control transcription and translation and whether or not it occurs. Just a couple of final notes to end this lecture. Proteins are very powerful things. In many disease states in humans and other animals, the cause of the disease is the overexpression of genes. In other words, too much transcription, too much translation, too much active protein. A lack of control, in other words. One of the best examples of a disease state that's brought about through the overexpression of genes is cancer. As we're going to talk about later in the semester, cancer is a disease where cells divide uncontrollably. And part of that uncontrolled cell division is the result of the overproduction of protein. It's not a question of whether or not a gene is present in a cell. It's a question of whether or not a gene is expressed that really matters. Is that gene turned on? Is it used to make an active protein? Remember, there are lots and lots of genes inside of your cells that will never be turned on as long as you live a healthy, normal existence. Proteins are powerful things, so cells must be able to control if a protein gets produced, when a protein gets produced, and how much of that protein gets produced.